Hello students, this is the first video of sexual reproduction in flowering plant, and topic covered in this video are structure of flower, stamen and its structure, structure of microsporangium and microsporogenesis. So let's start. Now let us study structure of flower. Flower is a shoot modified for reproduction. Flower bear reproductive organs. Stamen, which is the male reproductive organ, and carpel, which is the female reproductive organ. Stamen is formed by anther and filament, whereas carpel is formed by stigma, style, and ovary. Rule of stamen means group of stamens are called androecium, whereas rule of carpel is called gynoecium. Petal and sepals are non-essential organs of flower. Rule of petal is called corolla, whereas rule of sepal is called calyx. Rule means group. Now let us study structure of stamen. A typical stamen consists of long and slender stalk and terminal bilobed structure, that is anther. So stalk plus anther constitutes stamen. A typical anther is a bilobed structure, means anther has two lobes. This is the first lobe and this is the second lobe. In each lobe there are two theca. As you can see here, in each lobe there are two theca. So two theca in lobe 1 and two theca in lobe 2. So we call that anther is dithecous. And in each theca there is one microsporangium. In each theca there is one microsporangium, which is shown in red. In each theca there are one microsporangium. So there are two microsporangium in each lobe of anther. Now let us study structure of microsporangia or microsporangium. A typical microsporangium is circular in outline and is formed by four layers. The epidermis is the outermost layer. The endothecium is the second layer. Middle layer is the third layer, which is formed from 2 to 4 layers of cells. Tapetum is the fourth layer, which is formed by binucleated or multinucleated cell with dense cytoplasm. So four layers of microsporangium are epidermis, endothecium, middle layer, and tapetum. Apart from these layers, microsporangia also consists of connective tissue and sporogenous tissue. Now let us look at function of four layers of microsporangium. Outer three layers of microsporangium, that are epidermis, endothecium, and middle layer, helps in dehiscence of anther to release pollen grains. That is, these layers split open to release the pollen grain. While the innermost layer, that is tapetum, it nourishes the pollen grain. Now let us study microsporogenesis. We know microsporangia contains sporogenous tissue. Each cell of sporogenous tissue is a potential pollen mother cell. These pollen mother cells of sporogenous tissue undergo meiosis to form 4 haploid microspores. And this process of formation of microspores from pollen mother cell by meiosis is called microsporogenesis. These 4 haploid microspore change into pollen grain as anther matures, and the microsporangia forms pollen sac. So the 4 microspore will form the pollen grain, while the microsporangia will form pollen sac. So, a single microsporangia contains several thousand of pollen grains. So, if you have gained something from this video, please subscribe now to my channel Ramji Chopra, comment, like and share.